So I was asked to sort of give a perspective on my own career path and also the things that we do in our lab here at NIH. And so let me just introduce you to who I am. So as Rita said, my name is Kareem Zaghloul. I'm actually in the surgical neurology branch. And so in that branch, we are a group of neurosurgeons that both provide clinical care to patients here in the clinical center, focusing on different specialties and different patients with different types of neurosurgical disorders. But we also are each individuals who run their own labs and have their own research interests. And as much as possible, we try to align those two goals together. So many of my research interests are focused on data and issues that arise in the course of treatment of the patients that we see. I see patients largely who have epilepsy and also patients who have brain tumors, and a subset of those patients who are tumors that require mapping of function in the brain, and also patients who have tumors who have seizures. So what I thought I would do, and also let me also say at the outset, this is supposed to be pretty informal, so if you guys want to just interrupt and ask questions, that feel free. It's probably better. But what I thought I would do is sort of tell you a little bit about what we do and how we sort of merge these two worlds together in our lab and in our clinical practice, and then I'll tell you a little bit at the end about how I sort of got here and the steps that brought me to this point. And not that I have many words of wisdom, but maybe a couple sort of pointers that I thought were helpful on the way, that I thought might be helpful for some of you. And I know that there's a whole range of people here who are at various stages in their training and potentially have different interests in terms of what they want to do. And so I try to keep those points sort of as broad as possible. So I, as I told you, I sort of focus both on neuroscience and neurosurgery. It's a super exciting time in the neurosciences. As you guys probably know, there are many things that we are learning every single day that are giving us, sort of remarkable new insights into the brain, how the brain is structured, how the brain functions. We're learning things both at the molecular level as well as the larger sort of functional and systems level, how information is conveyed from one point to another between brain regions, and how, molecules and signaling pathways convey information within individual cells and between cells. And we're also now entering an era where we're actually starting to manipulate the brain directly. And so, there's optogenetics that's sort of been around now for, several years, but we also are developing ways, and our lab is actively involved in this process of using other approaches such as stimulation, electrical stimulation, to manipulate the brain. And the idea is, can we find a way to communicate with the brain? And so the reason that I sort of got into this field primarily is because I'm interested in understanding the brain, and I think it's a really fascinating problem. But the reason I got into part of the reason why I got into the clinical side of things that I do is because I think at the center of understanding how the human brain works is also human clinical neuroscience, which involves neurology, neurosurgery, psychiatry, treating patients with neurological disorders or psychiatric disorders. And so I think that plays a huge role and will continue to play an even larger role in understanding, how the human brain works and how we can move that understanding forward. So our lab, and like I said, I'm going to spend the first part of this talk just talking about what we do in our lab and some of the science behind what we do. And then I will go into some of the steps that brought me to this point. So our lab is focused on something called episodic memory. So as you guys know, human memory is quite an important aspect of human existence. In fact, all of your life you can think of in terms of memory. It really sort of allows you to think about you as an individual, about your past. It allows you to actually plan for the future because you base those plans on your memories of the past. And human memory is divided actually into several different types of memories. So there are things that are called implicit memories. These are types of memories like procedural memories, like learning or remembering how to ride a bike. You can't really explicitly describe these memories. They might be the memory of motor movements that you would need to ride such a bike or something like that. And so that's one set of memories, but then there's a whole other set of memories that are called declarative memories. And so these are basically, as the name would suggest, memories that you can declare. So this is, for instance, the memory, do you remember who the first president of the United States was, or do you remember who your birthday was? Or there's another type of declarative memory called episodic memory. And this is the memory of episodes or events that you've actually experienced at a specific time or place in the past. So this is the memory, for example, it could be the memory of your first date or of your last vacation. It's a memory of something that you explicitly experienced. And so there's a lot of research and a lot of people who've been studying how episodic memory formation works in the brain, and it seems to involve a complex interaction among multiple structures in the brain. So when you first experience an event, you process information about that experience in these cortical sensory areas and also in these cortical association areas. This is information regarding the items that you see in that event, but also the context of that event, like what room you were in or where you were in time or things like that. And all that information regarding an episode or an event's, specific information as well as the context within which it occurred is then conveyed down to structures in a part of the brain called the medial temporal lobe. These are a set of structures that are comprised of many different substructures, such as the entorhinal cortex and the parahippocampal gyrus. And these structures actually take this information about the information content of an episode as well as the context within which it occurs, and it conveys that down to this very important structure known as the hippocampus, which I'm sure all of you have heard about. And so the hippocampus has been clearly clearly implicated in memory formation. It's been obviously the largest sort of focus of research on memory. And the idea behind the hippocampus is that it takes all this information that it's receiving about an episode's, specific items and the context, and it integrates that into a cohesive memory. And then it actually communicates back with these MTL structures, with these cortical association areas, and also with the prefrontal cortex. And all of this allows you to experience an event and to encode the event and the details of that event into memory. Now, when you actually retrieve a memory, when you remember something, when you recall something, there's some evidence that suggests that actually what happens is that much of this activity that was present when you first experienced an event is actually replayed or reinstated. And this allows you to do something that you guys have all experienced yourselves, which is essentially to mentally and internally jump back in time and re-experience something that you had before had happened before. So you can think, for instance, of the first day you moved into college, and you could probably go back in time and re-experience that episode. And this is partly mediated by this ability of these structures here to replay some of that neural activity. So that's sort of what we do from a scientific standpoint, and I'll get into some details about that. But as I mentioned earlier, I'm also a clinician, and I work in the neurosurgery department, and many of the patients that we see are patients who have epilepsy. So epilepsy is quite a common disorder. It affects probably about 1% of the population. Epilepsy essentially is defined formally as having 2 unprovoked seizures. Basically, it is abnormal electrical activation in the brain that causes then some symptoms of some kind. And the symptoms that it causes depends on where that activation occurs. That abnormal seizure activity could be focal, so it could be constrained to a very small area of the brain and then just stop after a short period of time, or it could generalize and involve the entire brain. It could be seizure activity that can cause you just to have a very partial type of symptom, like for instance moving your hand, or it could cause you to lose awareness, for instance, and actually lose consciousness. And so obviously the first treatment for epilepsy in most patients is to give them medications, but it turns out that about a third of the patients continue to have seizures despite best medical therapy. So if you think about these numbers, so 1% of the population is about 3 million people here in the United States, and about 1 million of them continue to have seizures despite any type of medication. And generally, if they have tried 1 or 2 medications and they continue to have seizures, then the chances of them actually stopping their seizures from a third or fourth medication is basically less than 5%. And so this has a huge impact on people's quality of lives because, seizures, first of all, can be dangerous, right? So if you're driving and then you have a seizure and you pass out, you'll get into an accident. And actually, in most states, you can't drive if you have a history of seizures because of that. If you're swimming and you have a seizure, you could drown. But then it also has an impact on other aspects of your life. So in most places in this country, if you can't drive, it's very hard to actually get a job and maintain a job and consistently show up to work. And so that's an issue for people. A lot of these people obviously don't have jobs. And the other thing is continued seizure activity in the brain that can actually have a detrimental effect on cognitive function. So what you find in many of these patients is they have poor memory, they have poor executive function, and so on and so forth. So it turns out that a large majority of patients with epilepsy actually have seizures that involve many similar regions that have been implicated in this process of memory. And so these are temporal lobe regions of the brain. And so this is actually why one of the major comorbidities that we see in patients with epilepsy is poor memory. So what do you do with these patients who aren't responding to medication? Well, one option is to actually offer them surgery. And at first glance, this sounds kind of crazy, but the idea is that if we know in a particular person's brain that a seizure is coming from one particular spot in the brain, and then it's spreading to the rest of the brain, then the goal would be, okay, well, what if we go in and actually just remove that one spot? Can we stop the seizures? And it turns out that's actually that's a fairly successful strategy. But that strategy critically relies on our ability to identify that one spot in the brain. And so there are a lot of tools, diagnostic and clinical tools, that we use to actually figure out where that one spot is. So for instance, and the goal is to find this spot. We call this spot the epileptogenic zone. This is the area, this is a theoretical area, but the idea is this is the area that if we were to remove it, it would completely stop seizures. And so the goal is, how do we actually figure out where this epileptogenic zone is? And so we use all these different clues that we have from the patient's clinical workup. So for instance, if you're somebody who has seizures and it involves your hand, you're getting these hand movements, then there's a good probability that your seizures involve areas of the motor cortex or near areas of the motor cortex or are spreading to areas of the motor cortex. So that's one way to get a clue. Another way to get a clue is you can get EEG recordings, and you guys probably all know about these. But what we do is we'll bring patients into the hospital, we'll put scalp electrodes on them and watch them for a period of time, watch them have some seizures, figure out from the recordings where the seizures are coming from, and from there we can get a rough idea of what area of the brain is involved in seizure activity. Sometimes it doesn't give you a very specific answer, but it can at least potentially give you, let's say it's this side of the brain or maybe even this lobe of the brain. Another tool that we use preoperatively is MRI. So if everyone will get an MRI who has seizures, and in some of those cases, what we find is an abnormality on the MRI, a structural lesion of some kind. The most common thing that we see in patients with epilepsy are abnormalities of the hippocampus, actually, that structure that I was telling you about earlier in the context of memory. And if you see somebody who has seizures that seem both symptomatically to come from that side, from that scalp EEG to come from that side, and also has an MRI lesion that's implicated in that area of the brain, that hippocampal region, then you can make a pretty good guess that that's actually where those seizures are coming from, and you can make a pretty good argument to go in and take out that part of the brain. And so that's sort of the best case scenario. The cases that we see here are never like that. So what we see here actually mostly are patients who have incomplete information in one way or another. So we have oftentimes patients who have MRIs who don't have any abnormalities on their MRI, so we don't know for sure that there's something abnormal structurally that's causing these seizures. We have some patients who have maybe scalp EEG that would implicate one area of the brain and maybe symptoms that don't really correspond with that area of the brain, things like that. So there's a lot of instances where this information is discordant. It doesn't really line up. And so in those cases, the question is, can we actually do anything else to get more information? And so one of the things that we do in those cases is actually do a surgical procedure to actually put electrodes on the surface of the brain and actually deep into the brain. And the idea will be exactly like the electrodes that we talked about before, but in this case, these electrodes are now sitting directly on the surface of the brain, and we then place these electrodes. This is a picture from one of our surgeries. This is someone's temporal lobe. You can see these electrodes. These are these little metal discs. They're connected to these little wires that are coming out of their head. We're going to close all this up, and these little wires are coming out through the skin. It's a very sort of standard procedure that we do. But the point here is that it gives us actually very good precision, because now we are recording from each one of these little individual brain areas. We can, after this patient has completed their surgery, we can watch them here in the hospital for a week or 2 weeks or 3 weeks. We can wait for them to have some seizures, and we can see what that activity in the brain looks like when they have their seizures. And this gives us a very precise picture of where their seizure activity is coming from. And so we do this all the time. We have somebody who's here right now doing it. And so this is sort of a very, very powerful clinical tool to allow us to figure out where someone's seizures are coming from. But it turns out it's actually a very good research opportunity as well, because as you can imagine, we're recording signals directly from the surface of the brain while these patients are awake and they're here in the hospital, and they're basically just sitting around. And in the first couple days, they are a little bit, uncomfortable and recovering from surgery, but after that, they're kind of just bored, and they're watching TV and they're just reading, and basically they're willing to participate in different types of games or tasks that we can design to actually look at the function of the brain. And so this gives us a very powerful way to look at sort of signals in the brain, neural signals in the brain across multiple different spatial scales. So from any one of these electrodes, we can record the activity over this small region of the brain. This gives us a sense of the local activity of the brain. We capture these signals that are basically these intracranial EEG signals or local field potentials. You can see that they're characterized oftentimes by the presence of these fluctuations or what are called oscillations. These oscillations have been very well described in literature. They come in a variety of different flavors or frequency bands, and they've been implicated in a lot of different types of functions, cognitive functions like movement or thinking or attention or memory. We can also look at what happens when we look at the activity across multiple areas of the brain and how that activity is coordinated between these regions. And so this allows us to think about the brain at a larger level, at this level of networks, of this information that's being conveyed from one point to the other. And allows us to approach the same spatial scales that we think about when we use other modalities such as functional MRI, or even when we look at things like PET imaging, or even what happens when you have a lesion or a stroke. And then more recently, we've sort of developed the ability as a field to record now the activity of individual neurons. So we can actually implant little microwires directly into the brain. Each of these microwires sits next to one of these neurons. It captures action potentials, and we can ask what these action potentials are actually doing. And so now what you have here is an experimental platform. Besides obviously the clinical utility of this, you have an experimental platform where you have an awake human person where you're looking at neural signals across all of these three sort of scales of spatial resolution. And you can ask what neural activity is doing as people are doing different things that you ask them to do. And so if you think about, our goals in neuroscience, which is to understand what the brain is doing and what these different brain regions are doing, we have a lot of different tools in the neurosciences that allow us to approach that goal. So we have things like functional MRI, we have, TMS, we have PET imaging at a very smaller spatial scale. We have, single unit recordings and multi-unit recordings from, dish, neurons and culture or whatever. And you can see that each of these different tools has this trade-off between temporal resolution and spatial resolution. And if we see where these human intracranial recordings lie in this spectrum, we see that it gives us a very powerful way to examine what it is that the brain's doing. And so we think this is a great opportunity. So in our lab, as I told you earlier, I gave you that whole introduction about memory and what we think about memory. I'll show you just some simple example of how we do this to actually understand what happens when you actually form and retrieve memories. And so one of the things that we do quite frequently is we have these subjects or these patients play these little games. And so this game is a game where we give them pairs of these words and we ask them to form these novel associations between these words. It's called a paired associates task. Then they do some math problems just to distract them for a little bit of time. And then later on we cue them with one of the words, and their job is to remember what the associated word is. And we do this while recording activity from each of these electrodes that we've implanted. This is a reconstruction of one of our patients with the electrodes on their temporal lobe. And so from each of these electrodes, we can see what the activity looks like during each one of these individual trials. And we can divide those trials into the ones that they were able to remember and the ones that they were not able to remember. And we can ask if there are any differences in the activity that we observe. And if we do this across enough people, and we do this enough times, we actually can so if we do this across enough people and across times, we can actually get these very, very high spatially precise maps and temporally precise maps of what the activity in the brain is looking like when you're remembering. And so what we're looking at here is a little sliding window of what happens when the word comes on the screen, which is indicated by this dashed line over here. And what we're looking at is the activity in brain regions, and this is average across multiple subjects. And what it's saying is that when the word comes on the screen, if you're going to remember that word, you have this activity that starts, this high frequency activity that starts in the back of the temporal lobe over here, and then migrates to this other brain region called the dorsolateral prefrontal cortex. So we can get very, very, very nice maps and pictures and a good sense of what's happening in the brain when you are doing this. And so just to give you a more specific example using some of these other electrodes that we have, which are these microelectrodes. So these are the electrodes that I mentioned earlier. These are little arrays of very, very, very small electrodes. They are about 400 nanometers apart. The whole thing is about 4 by 4 millimeters, and so that's how big that thing is with respect to a penny. This thing extends about 1 millimeter, and so these are all little electrodes that are extending 1 millimeter, and we extend that, we put that directly into the brain. So it's extending 1 millimeter into the brain, and it's recording from each of these electrodes the neural activity around that electrode contact. And so the hope would be that we can actually capture some neuron activity around each of those contacts, so we can capture action potentials. And so just to show you an example of how this works, so what we did is we looked at, for instance, activity when patients were performing this task. Again, this is the same paired associates task where we ask them to remember these pairs of words, and then we cue them with one word, and their job is to remember what the associated word is. And we did this while looking at this activity in this region of the temporal lobe called the middle temporal gyrus. And the reason we picked this region is because when you perform this task, it requires you to know what the meanings of these words are. Like if I tell you to remember that there is an association that you're trying to form between beam and camel, you're not going to make a new word called beam camel. You're probably going to think of a picture, for instance, in your head of maybe a camel standing on a beam or something like that. But it requires you to draw upon the meanings of these words, and this requires what's known as semantic processing. And the evidence for that we have for that is if you actually look at these word pairs that they were able to remember correctly versus the ones that were not, what we find is that the ones that were correctly remembered tend to be more semantically related. They tend to be more similar to one another than the ones that were not. You can imagine that it might be easier to remember bread butter than bread shoe. And so semantic processing involves many, many areas of the brain, but one of the core regions of semantic processing is actually this temporal lobe region. And so we can see what this activity is doing in this region of the brain. And so we implanted these electrodes in these patients, and so just to show you what these data look like, what we're looking at here are all these individual neurons from one subject in one experiment, and this is what happens during time after the words come on the screen. And the color tells you what each of these neurons is doing when the words come on the screen. And this is what's happening when they're studying this word pair. And you can see that some neurons are increasing their activity and some neurons are decreasing their activity. But interestingly, if you look at what happens during retrieval, when they're remembering this activity, you see a very similar pattern of activity. This dashed line marks when they actually vocalize their response. And you can see the same neurons that were showing increases in activity during encoding are also increasing during retrieval. Conversely, those that are decreasing are decreasing. And so this suggests what we had speculated earlier, which is that when you encode something into your memory, there's a pattern of activity that exists in your brain. And then when you retrieve memory, you mentally and internally jump back in time and you essentially replay that pattern so you can re-experience that event in your head. And so this is giving us evidence that this actually happens. So that's sort of my quick summary of what we do. There's obviously a lot more about what we do, but I thought I'd spend the last few minutes sort of talking about my own career path, which is, I know, something that Rita had asked us to talk about. And sort of some of the, like I said, some of the little pointers that I thought were relevant along the way. So I get asked all the time from students, like, how do you plan to do this, and how do you sort of, what are you supposed to do to actually get to, this goal? And it's funny because I don't actually have a very good answer for that question, because my answer is that I had no plan. Like, I basically just did what I thought was interesting at the moment, and I didn't really think that, I was going to do this specifically to get to this goal, specifically to get this goal. Instead, I sort of did what I thought was the most interesting thing. And so at the time that I was in high school, I didn't really think at all about medicine or biology or science or neuroscience at all. I was thinking more about math and physics, because that's sort of the things that I was interested in, the things that I like to do. And I actually ended up going to school at MIT, where I actually majored in electrical engineering and computer science, so something completely different than sort of biology and the neural fields. And I didn't really know anybody in my family who was a doctor. I didn't really know anybody in my family who was doing biological sciences. And so this is sort of what I thought I was going to do. And I did it because I thought that was the most interesting thing. But while I was there, I ended up actually stumbling into some courses and some professors who were thinking about how you can use sort of principles of electrical engineering to think about the nervous system, because the nervous system ultimately is an electrical circuit. And so you can think about how you can model the nervous system in this way that you think about electrical circuits. And so I thought that was interesting, and I thought I would sort of pursue that. And I thought that I was going to end up going to graduate school for that in terms of doing a PhD. And like I said, I don't know what your career paths or your plans or your ideas or aspirations are going to be. And I'm sure some of you are thinking about medical school and some of you are thinking about graduate school. I'm sure some of you are thinking about both, and maybe some of you are thinking about neither. But so what I was doing at this point was thinking about graduate school. But I started thinking about this idea of sort of, neuroscience and thinking about this cross between electrical engineering and neuroscience. And then I thought, well, it might be interesting to combine this somehow with medicine. And I didn't really have very clear reasons for that at the moment. I thought it was an interesting idea. It was sort of these naive thoughts that, number one, I thought it'd be great to help people, which it turns out that it is. And number two, that I thought it would be great to study the body, because I thought the body was a very well-engineered system, and I thought that'd be really interesting to learn about. So I ended up actually going to MD PhD at Penn. And one of the things that sort of was interesting at the time that I was applying there was not many people who were coming from a computational or engineering background who were applying to medical school at the time. I think it's changed a lot since that time, and I think medicine in general has been a lot more open and a lot more embracing of these computational approaches. But at the time, that was certainly not the case. But the takeaway that I sort of took from that experience was that you shouldn't be afraid to cut across disciplines. That if you are learning something in one discipline, you could use that, and it actually gives you tremendous insights into other disciplines. And if you're working within a discipline, you should look to other areas for inspiration, because there are oftentimes problems that are quite analogous that have been solved by other people in a different way in a different field. But this idea of sort of merging across different disciplines is actually at least for me, I thought has been actually quite helpful. So I ended up going to Penn, and I did my MD and my PhD there. Like I said, I was primarily interested at that time in doing a PhD. I was interested in the medical side of things for those two reasons that I talked about earlier, and I just wanted to see how it would go and what would happen with it. But like I said, I wasn't really planning at that point to do neurosurgery and have a neuroscience lab and end up at NIH. I was kind of just doing what I thought was interesting at the time. One thing that happened to me when I was at Penn, and this is sort of the takeaway from this point, was I got very lucky, because when I started there at Penn, I ended up taking a year off and actually going to study history and literature and then coming back. And in the time that I was gone, they ended up hiring this young assistant professor who just started there by the name of Kwabena Boahen. And he was a guy who came from Caltech, and his whole thing was, he was actually a student of a guy named Carver Mead, who you probably haven't heard of. Carver Mead is actually a big deal in the field of developing integrated circuit chips. And so their big thing was, can you develop these chips and build these chips in a way that actually models what the nervous system is doing? And can we use these silicon chips? The way we use silicon chips today is, we used everything digitally. So everything's either a 1 or a 0. You drive everything all the way up or all the way down. But there's a whole range of sort of, analog signals between those 1s and 0s that you can use to actually model the circuitry of the nervous system. And so their idea was, can we do this to model neural circuits? And so when he showed up at Penn, I thought that was really actually quite interesting, and I immediately joined his lab, even though he didn't have a lab at the time. And our project was to model what the retina does, what the visual system does. And so this is a circuit diagram of the retina. Light comes in here, it activates these rods and cones. This information is conveyed to the bipolar cells and then ultimately to the ganglion cells that actually make up your optic nerve. But then there's some interactions that occur in these different layers between the photoreceptors and the horizontal cells and the bipolar cells, and then between the bipolar cells, the amacrine cells, and the ganglion cells. And so we ended up actually modeling each of these interactions using these transistors. We built this little silicon circuit that sort of does what we thought the retina does. It's sort of main stages of processing. And so just to give you an example of how that works, if you look at, for instance, a moving grating, and you present that in front of the retina that we built for my PhD thesis, what you find is that you can have these different responses from different, what we call neuronal subtypes. So you have neurons that respond to when the grating comes on, neurons that respond to when the gratings come off, neurons that respond transiently, neurons that respond sort of more persistently. And so this is sort of an example of this whole array of neurons responding to my face and me moving in one direction or another. So that's sort of what I did. But again, it was something where I didn't really plan, that he was going to be there. I didn't really know what I was going to do my PhD in, but I found this person, and it was a great opportunity. And immediately I said, okay, well, this sort of aligns with the things that I like. I don't know where it's going to end up, but I just want to do it, and we'll see where it goes. So at the end of that, I was sort of trying to figure out what to do. Obviously, I had a couple of different options. One option was to continue doing sort of postdoc research work, or even to go work in a company. Another option was to go into residency, and the obvious choice in that point was to do a residency in ophthalmology, since this is all sort of visual related in retina, and to develop these sort of artificial retinas. But I ended up actually liking neurosurgery, and that's a whole other discussion. I ended up doing that. And in neurosurgery residencies here in the United States, most programs actually dedicate a set of time for you, about 2 years, to do your research. And I, again, was once again fortunate enough, because I didn't know who I was going to do my research with, but there was a guy who actually also just came to Penn after I started my residency, named Mike Kahana, and he was looking at how we can use the signals that we capture from these neurosurgical procedures to understand what's happening in the brain. And so the takeaway that I took from actually joining Mike's lab, and actually had a great time in Mike's lab, was I didn't didn't allow the visual processing work that I had done in my PhD to constrain what I was going to do moving forward. I certainly used it to inform sort of the types of analyses and the types of things that I learned from that, but it doesn't constrain you, it should inform you, and that should be true of all of your experiences. So whatever you're doing now is not necessarily what you're going to be doing 5 years from now or 10 years from now, but you can learn from what you're doing now and use that in the future. In Mike's lab, we principally were looking at activity both in the brain and the cortical regions as far as memory, but also in these subcortical regions that are responsible for dopaminergic signaling, like the substantia nigra, and how those signals change when you are, let's say, in this case, playing this little task where you are choosing a deck of cards, and in some choices you get a reward, in some choices you don't. And what we showed was that these dopaminergic neurons show these phasic responses. This is very analogous to what we've seen in the animal literature. The last piece of advice is, or the second to last piece of advice was, obviously you need a team, you need those collaborators and friends and colleagues to make this work, and that's probably the most important thing. Everything that we do in our lab is quite collaborative within the lab, but everything that I've been able to do over everything in my life, I think, is largely due to the people that I've been working with, and I've had very fortunate and good relations, and so I think that's sort of something that's fairly understated, but I think it's a super important point. And then finally, I know a lot of you were asking about work-life balance issues, and I don't know if that there is a clear answer or a single answer to that. This is my work-life balance issue. These are my 3 kids, and so obviously I spend a lot of time trying to make sure that I'm hanging out with them and doing stuff with them, and they are a lot of fun and sometimes a lot of work, but they're mostly a lot of fun. But there isn't a single answer, and I think the best answer I heard was at another talk, or symposium I went to a while ago, and the woman got up there and said, look, everyone's got to make their own decisions about what they think is important and what they don't think is important, and what you're going to sacrifice and what you're not going to sacrifice. And I've made those decisions for myself in some ways, and what I'm definitely going to do for work and what I'm definitely going to do for them, and I think that just depends on every individual person. So I don't think there is a single answer. So that's it. These are the members of my lab, and thanks a lot. Any questions? Yeah.
This is about the surgeries, removing the, what was it called? Epileptogenic.
Mm-hmm.
So do patients ever experience any adverse effects after you remove their brain?
So first of all, there's always a risk of complications from surgery. So if a complication happens, then yes, they will experience adverse effects. But in the vast majority of cases, those complications don't happen. However, even in cases when you don't have complications, when you're taking out a part of the brain, it can affect them in different ways. And so especially when you're taking out structures in the temporal lobe, like the medial temporal lobe structures, like the hippocampus, that can have an effect on their memory. For example. And it turns out it's really hard to predict what kind of effect it's going to have on someone's memory. That's one of the things that we're thinking about and trying to figure out how we can do that. But one of the sort of things that we've noticed is the people who generally have very good memory going into a surgery feel a much larger difference in their memory after surgery than the people who have a very poor memory. And that makes sense, because if you have a very poor memory, you don't really have much to change. There's going to be much that it's going to change after that. So, yeah. So we try to place those penetrating electrodes, those microelectrodes, into regions of the brain that we think we're ultimately going to remove. And so I think we've done this, I think, in, I don't know how many patients, but the majority of those patients, we've actually removed those areas of the brain. There are a couple of patients in whom, when we had placed the electrodes, they either didn't have seizures during the entire time, or their seizures weren't really specific to a single location, and we couldn't do a surgery, and so we had to take those electrodes out. It doesn't seem like there's anything that's changed because of those electrodes, but to answer that question, we'd have to wait like 10 or 15 years and see what happens. Yeah. Yeah, that's a really good question. So we haven't actually addressed that particular point. So there are a lot of sort of databases out there that sort of can tell you how semantically related different words are, and there are a lot of different axes upon which words can be similar. So they could be similar because they actually look similar, like brick and block, for instance, or they could sound similar, or they could have similar meanings. And so there's a lot of different axes. But you're right, probably most of those databases and most of these sort of similarity scores are based probably on Western English language culture. So we just haven't really explored that point. Any other questions? Yeah.
Do you allow summer students to watch the surgeries?
So this is a question that I have tried to refer to all of these people, because here's the deal. We used to have summer students or all students come to our surgeries all the time. And I love having students in our surgeries all the time. And I literally had students in my OR almost every single case that we did. And then about a year and a half ago or 2 years ago, NIH decided that they are no longer to allow students in the operating room. And so I have been appealing to our NINDS leadership, to many, many people, to allow that to get that changed, but it has not happened.
Yeah, we have not been super successful. So the NIH, the whole clinical center underwent a review that didn't go as swimmingly as everyone had hoped. And a lot of the policies got tightened up. And one of them was having students watch surgeries, or even students that are older watch surgeries for research that they are not directly involved in. So if you were directly involved, last year we could make the justification that if you were directly involved and it was important to your research. And we got like maybe 1 or 2 in. I mean, we think it's so important that we'd love for everybody to see it. I mean, it's an amazing thing if you're good with blood. But, you know, it's an amazing thing. It's fantastic. So yes, we are trying to figure out a way to make it work right now. It is not up to us. If it were, everybody would be doing surgery right here.
Yeah. So, I mean, from my perspective, I have no problem with it, and I would love for you guys to come. I actually think, first of all, one of the core missions of NIH is training, and so this is actually training. And then for me, from my own personal experience, the only reason that I even knew about neurosurgery is because I had an opportunity like that. And so I think it's a very formative experience for many, many people, and I think it would be great to. I leave it in Rita's hands. Thanks.